What if I told you that even today, we develop drugs using models that scientists already know don't reliably predict human response? Now, what models am I referring to? Well, not just the animal models like rats or mice or rabbits that popped into your head, but even cells that are grown in petri dishes. What do I mean by that? Well, don't worry, we're going to be discussing that in today's installment. But before that, I want you to have one major question stuck in your head, which is, we've spent decades, billions of dollars, and millions of animal lives on the cause of drug development. Yet, most models fail when they reach humans. So, what exactly is going wrong? What exactly is broken in this process? Well, in today's episode, we'll figure that out together. Modern drug development relies on two main testing methods before the drug even reaches the market. The first method is growing cells in a petri dish, what scientists refer to as 2D cell culture. The second method is animal testing, commonly done on mice, rats, or rabbits. Now, the goal of these two methods is to reliably predict human response to a certain drug. But the uncomfortable truth that we're starting to find out is that neither of these methods does a very good job at that. Let's start with why 2D cell culture models fail to reliably predict human response. In 2D cell culture models, cells are grown flat. They're either stuck to the surface of the container, or isolated, or soaked in artificial media. There is no blood flow, no immune system signaling, no mechanical forces, or even communication between other cells/tissues. This is not representative of how cells live inside our body. In fact, many studies show today that conventional 2D cell culture models are poor predictors of human response. On a scale from 0 to 10, with 10 being the most predictive of human response, traditional 2D cell culture models don't even make it halfway through. So, what do we do next, given that 2D cell culture models aren't reliable predictors of human response? We moved to animals. Mice, rats, rabbits, sometimes even dogs or monkeys. But what we need to understand is that animals aren't tiny humans either. Granted that the mice share most of our genes, we are still biologically very fundamentally different from the mouse in terms of our metabolism, our disease progression, hormonal regulation, and immune responses. For these reasons, animals are also poor predictors of human response to a certain drug. Is it better than flat cells? Yes. But is it still reliable? Nope. Well, if animal tests fail us so often, why are we still using them? The answer to that is habit, regulation, history. It is often easier to keep doing what we know, even if it doesn't work out well for us. And this mismatch in predicting real human response by existing models in the drug discovery space has its consequences. According to a 2023 survey, over 92% of drugs that looked promising in preclinical trials failed when it came to human trials. Some of them simply did not work in humans. Others turned out to be toxic, which means that these useless drugs are being progressed forward in the drug discovery chain, while potentially life-saving ones are being held back. All of this at an enormous cost. It takes 10 years and $3 billion to get a single drug out into the market. This is simply not only inefficient, it's absolutely unsustainable. And on a more serious note, behind every failed drug is a person waiting. Someone with cancer, someone with Parkinson's, someone with Alzheimer's or endometriosis. These are complex, multi-organ, and deeply human diseases, exactly the kind of conditions that flat 2D cells or animal models struggle to replicate. If we don't have models that replicate real human biology, how can we expect human relevant results? And this is exactly why scientists are looking for alternatives to drug testing. Not just because animal testing is costly or ethically questionable, it is because the data often fails to directly translate to humans. It's simply not just activism, it's scientific consensus. So, if 2D cell culture models don't really work, and animal models are not good enough for drug testing, then what does work? Well, here's the exciting part. Scientists are developing new models that combine the strengths of real human cells in real biological environments with real-time behavior. What do I mean by that? Well, models that pump, breathe, or flow even. Sounds like science fiction? Well, it's not. In the next episode of the series, we will be diving deep into the breakthrough technology that is organ on a chip. So, in the meantime, like and subscribe for more content. It is futuristic, it is a little freaky, but it's absolutely real. I can't wait to see you in the next episode.